Hello and welcome to PK Tech Lab. Today, we are going to dive into the Excel Home tab with examples geared specifically towards scientists and lab technologists. Whether you are formatting experimental data, managing lab results, or calculating scientific trends, we'll show you how to use Excel Home tab efficiently. So let's get started. The Home tab is where you'll find all the essential tools you need to format and organize your data, from modifying text and numbers to sorting and filtering results. The Home tab helps make your scientific data more readable and easier to analyze. We'll start with the clipboard group, and let me show you the clipboard group on the screen. Okay, so this is the clipboard group. This is the clipboard group. So the clipboard group is made up of the paste command, the cut command, the copy command, and then the format painter. This is the format painter. So this is copy, this is cut, this is paste, and then the format painter. So now let's talk about each one of them. So the copy and paste command. So let's say you have a data set like what you see here. So we have a data set of concentration against absorbance. You have a data set of concentration against absorbance. And then you want to copy this data and paste into a different cell or onto a different sheet. So you can select data, click on the copy command here, select the cell in which we want to paste the data, and then we click the paste command here, and we have what we selected in our new location. So that is how the copy paste command is used. Okay, so this is great when you want to separate raw data from processed data. And so let me explain that. Let's say this is the data you got from our analysis. So let me add sample names to it. So we have sample A, sample B, C, D, E, F, G, H, I, and J. So let's say these are the sample names, okay? So let's bold in this, okay? So we have this, and then we want to perform our analysis. We can open a new sheet and then copy this data that we have here. We'll copy the data that we have here and then paste it into the new sheet. We'll paste the data here and then perform our analysis on this new data so that in case we have any problems with or there are some changes to this data during analysis, it will not affect the original data that we started with. So that is why sometimes we might need to cut or copy and paste data into a new worksheet before analysis to avoid losing our data. Okay. All right. So that is it for the copy and paste. Now cut and paste. When we cut and we paste, we lose the data at the original location where we cut the data from. So for example, with this data, if we select all the data sets and then we cut using the cut command and paste into a new location, we lose the data that we have in the original location. So it's like cutting a part of a paper out of a book, you are not going to get the information that was on that sheet when we go back to that. Okay. So when we are cutting and pasting, we need to be careful when using that command because it's possible to lose your data while you cut and paste. All right. So we've cut and we've pasted our command using the clipboard. So those are the things that we can actually do with the clipboard. Now, I'd like to mention that the commands in the clipboard, we can also simply locate the command by right-clicking on our sample, select the sample, and then we have the cut, copy, paste commands in the pop-up menu that we can also use to quickly cut, copy, and paste. And then there are keyboard shortcuts that we can also use. For the cut, Control C for copy, Control C for cut, Control X, and then for paste, Control V. So those are different ways that we can actually cut, copy, and paste our data while working with Excel. Now let's talk about the Format Painter. Format Painter is especially useful when you are working with multiple tables. So we have, let's say this is table. Let me copy this table. And then paste it here. Okay. And then let's say we edit this table and we want to replicate the editing that we did here in another location on the page. So you formatted this concentration absorbance to be bold, and then you want to replicate this formatting in another header. You use the Format Painter to copy that formatting and apply it to other tables. So let me label this PH. And then let me add another data. Let's say, okay, so we have absorbance, then we have concentration, we have pH of the sample. Okay, so let's work with the pH. We can click on this, select Format Painter, and then paste it here, and our pH is bolding because the formatting that we selected was bold. You get, so we don't need to go back, select, and click bold, no. But let's put another, okay, let's do the same thing for the sample header. So absorbance is boldened. We take Format Painter and we apply it to sample. So you realize that sample has been boldened and centered because absorbance is bold and centered. You understand? So we are not now going to select. Let me undo that. If we are to do that manually, we are now going to select sample, choose bold to bolden it, bold to bolden it, and then we go to align center, to center align it. All right. And if we wanted it to be in the middle, we would have to come here and select. So there are 3 different steps we have taken right now to get the sample header to look like the others. But let's say we've done that already for the absorbance and we only want to replicate that. So let me undo again and repeat what I did. You just select your absorbance that has all the formatting that you want to apply to your sample, and then select your Format Painter and you apply to sample and it is done. So that is a fast way to actually format similar headers or cells that you want to apply similar formatting to. That is how you can do that. So that's it with the format painter. And that is it with the clipboard group. So we've learned clipboard group contains the paste, copy, cut, and format painter commands. So now let's talk about the font group. Let's talk about the font group. So this is a font here. This is the font. Okay, let's redo that. This is the font. Yes, so this is the font group. So let's talk about the font group. So we'll explore the font group where you can change how your text looks. This is important for making your data tables and graphs stand out in lab reports or publications. So we have different commands there. We'll start with the bold, italic, and underline commands. Okay. So for example, you have this data that is showing on your screen from a lab experiment, right? You can highlight the headers like concentration, absorbance, and sample and make them bold. Already what we have is already bold. So let's undo that and re-demonstrate what we are seeing. So same way you can choose bold. When your text is already bolding, selecting bold, remove the bold formatting. So we have it selected, we want to bolden it, we choose bold, and our text is boldened. Let's say we want to italize it. We check the italic command, and then the text is italicized. All right. What if we want to underline it? So the formatting names are very descriptive and they do exactly what they are named as. Okay. But this underline command, you could choose different ways you want to underline your text. So what I have here is underline. This is a single line, and then we have a double line underline here. You can actually make those, choose those different commands to get different results. All right, so let's bold in our text. Let's eliminate underline. And that is it for the bold, underline, then italic commands. So font size, font color. What we have here, the size or the font size is showing in the font size pane here. All right, so at the moment, the size of our header is 11. We can change that. Let's say we want it to be bigger, so 14. Right. The font type we have here is Calibri. Let's say we want to change that to Times New Roman. We have it here. You can actually search, and then we have the font type changed to Times New Roman. So that is how we can format the font type and then the font sizes using the font, right? Using the font. But my header color is black. The text color is black. I want it to be, say, yellow. I can go to the font color command and then change that to the color I desire, right? And then my background has no fill at the moment. but then I want the background to be blue. Okay, so I can go to the fill color and change the color of my background to blue. Now, this is a point where you might very well understand the function of the format painter better. So let's say we run an experiment and then we have results for the pH and then the electrical conductivity. But then we want these two headers to have the formatting of this header that we have, these headers that we have here right now. We can select this, click on the format painter, and then the formatting here has been copied. We apply that here, and as you can see, the formatting has been applied to these headers too. So format painter can really be helpful. So we've talked about the fill. Borders. You might want to thicken the borders of your header. So let's select this headers here and then say outside borders. Okay. Outside borders. Let's choose thick outside so that it will be more visible. And then you can clearly see that the border has been applied to the header. Border has been applied to the header. So that is the function of the border. You might not want borders at all. And then you select this, no borders, and then the command is applied appropriately. So the borders have been eliminated. So that is it for the font group, the home tab. Now let's talk about the alignment group. When you are working with scientific data, keeping it neat and easy to read is essential. So for example, you have these data points as is showing on your screen right now. Normally, when you type in the data without any formatting, this is how the text will look. But then maybe you want to keep your headings centered. So you go to the alignment group and then you select center align as it's showing here. So center your content, center align, and your data or heading is centered. Let's say you want to right align the data points under concentration and absorbance. You select all your data points, you go to alignment group, and then you select right align or align right. And then the data is right aligned. So that is how we can use the alignment group to format our data. These two are indentations. Maybe you want to indent to the left or to the right. Okay. You can use these commands for that. All right. And most of the time, we wouldn't like to use this, but when you need to orient your text diagonally, basically you can use this command. But now I want us to talk about the merge and center command. So let me select these two cells and then put a border around them so that they can be very visible to you. Okay, so you can see that these two cells are unique. I want to merge these two cells so that, let's say I have a text inside, say concentration of ammonia. You would see that this wouldn't fit into one cell. Let's say I want to keep my data points into the cell size as they are. What I can do is to merge these two cells together. Or in this case, there would even be three. Go to align group, merge, and then I have enough space to contain a heading that is as long as this without altering the cell sizes beneath it or above it. All right. So this becomes essential when you want to have, let me, not gotten there, but let me insert. Okay. When you want to have a heading for all your samples. So this one is a collection. So this data point that we have here, say you run a test to determine, this is a test to draw a standard curve. So standard curve for, say, sample. Yeah. Okay. You want to keep it above this data point without changing the formatting of these data points. What you can do is to select the cells, merge and center, and then format your cell, your heading to look the way you want it to. We've spoken about these ones already, so I wouldn't touch on them. 16, right? And then I can fill with the color of my choice. Okay. So this is how you might apply merge and center, and in different situations, you might use it differently. But the point is that it merges two or more cells together. But then you have the text as shown here, concentration of ammonia, but you don't want it to cover several cells like we have done. You want it to be in this one cell, but you don't want it to cross over to other cells. What can you do? That is the function of the text. So you see that it is wrapped into one cell so that it doesn't overflow into other cells and interfere. So the wrap text will do that for you, and it is found in the alignment group. So that is it for the alignment group. That is it for the alignment group. So let's move on to the next group, which is the number group. The number group. Okay. So the number group is shown here. Like I said, my screen is small, so but then when you click on it, everything that is supposed to show will show. For you with maybe a widened screen, see the commands appear on your screen. All the commands appear like how the font group is showing here. Okay. So the number group, as scientists, what can we use that for? We can format our data based on its type. And so this is critical in science where data precision matters. Okay. Before we continue, let me delete this text here. All right. So let's say you're working with a set of pH measurements. So let's type in some pH measurement. So 3.3, let's say 3.0, 4.89, we have 7.8. And then we have 14. Now let's see, 13.3. Okay. So you're working with a set of pH measurements. And you can format the data to display a specific number of decimal places, ensuring accuracy. So how can we do that? We select our data, we go to the number group, and then this one here, we used to increase decimal places. This one here, we used to decrease. And The decrease will round up. The increase will add more places, decimal places to the number. So this one is showing mainly 2 decimal places, but I want to increase all of them to a decimal place of 4. All right, so we have 3 here, and then 4 decimal places. You can do that. Let's say we want to reduce it to 2. We have that here, to 1, and that is how these two works. Okay. All right. Now we can also use scientific notations to display our values. So we have a set of observances here, and we want to display it in scientific notation. So we go to the numbers group, we click on the arrow, drop down arrow icon, we select scientific, and then our results are displayed in scientific notation. But we don't want that right now, so let's go back to general. But that is how it can be done. But say your table, instead of being table of results for a scientific experiment, you rather have a table for budgeting for lab supplies. And so you want to format your table to work with currencies. You can also do that. Go to the numbers, and then we have currency. So a dollar sign has been applied to each and every one of it. So let's recap on the numbers. So in the numbers group, we have several data formats that we can apply to. General takes both text and numbers, so alphanumeric data. Number would specifically work with numbers. Currency will work with numbers. Counting will work with numbers. Date, time, percentage, fraction, scientific notation, and then text will take text. All right, so that is it with numbers group. Lastly, let's discuss the editing group and explore its features. So let me highlight the editing group. Okay, so we have the editing group here. We click on it, we see that the editing group is made up of AutoSum, fill, clear, sort and filter, find and select. So we would explore the function of each. So sorting and filtering, you're working with large data sets and you need to sort them maybe either in ascending order or descending order, or even custom sorting. You can use the sort and filter command. So see that the first one is sort A to Z. This is ascending. Sort Z to A. This is descending. Custom sort. You define the terms of your sorting. And then the filter command here is for filtering. That is narrowing your data to find information that you want easily. So this is our data set. Let's say we want to sort this data in descending order, Z to A. Okay, so what do we have? Wait, before we do that, let me change this back to, but it should actually not affect the sorting. So sort and filter Z to A. Yeah, and we have this one in order. Okay, let me change this one back to B so that you understand what I'm saying better. So we select and we sort Z to A, and it's clearly saying that the arrangement of the data set has been reversed, starting from J to A. All right. If we want to take it back, sort A to Z, and then we have it. But then, let's say you only want to look at certain samples in your result. You want to go to sort and filter, and then select filter. You see that a filter has been applied to each data group. But let me do this in a better way. So let's remove the filter and then apply it instead to the headers. So add filters, and the filters have been applied to the headers. So let's say I want to only view sample D. So first you see that all the samples are selected, but now I want to only view sample D and I. Sorry, yes, I. So I select sample D and I, click OK, and then only D and I appear with their concentration and absorbance, right? So this is how you can use sorting and filtering to manipulate your data for the outcome that you want. Let's undo what we have done, and then let's talk about find and replace. Find and replace. So we go to the editing group again. We have find and select command here. On some versions, it is called find and replace. So we select our data set. Let's say I want to find sample B and rename it as standard B. We find using the find. No, we find, because we want to find and replace, we use the replace command. So what do we want to find? We want to find B. What do we want to replace it with? We want to replace it with standard B. So we replace all, if we want to replace all data points, all sample B's with standard B, we select replace one. And you see all done. We have made 3 replacements. Okay. So we go back to our data and then we find that, all right, yeah, we find that standard B has been changed to B. Standard B has been changed to B has been changed to standard B. So that is how the find and replace works. And that is how it can help us go around our data. All right, so you would observe something. They said 3 replacements made. Take a look at this cell. This cell had the name absorbance. All the B in the spelling has been changed to standard B. Actually, this is not what we want. So let's try again and see how we can bypass that. So instead of selecting with the header, we only select the data points like this. We select the data point, go to editing, find the replace B with standard B, and then replace, find what B, replace, yes, and one replacement made, okay. So the absorbance is not affected. It's because we selected it and we chose replace all. But in this case, we only selected our data. All right. So we want to be careful when we are editing our samples. So when we are editing our data, so that we don't change things that will affect our data in the end. And that is why it is important to copy our data into a new sheet. So we can name sheet 1. We can rename it as working sheet. And then we name sheet 2 as original data. So that if anything goes on with the data we are working with, we can easily go back and reference to ensure that we are not mixing up stuff. So that is the way to find and replace. We will normally not add concentrations together or absorbances together, but for the sake of demonstration, let's demonstrate the AutoSum function, how it works. So the AutoSum here has sum, average, count numbers, max, min functions. There are more, but I want to only use the sum. So we want to sum all the absorbances that we have here. We go to AutoSum. We see that it is automatically selected. We end, then the sum of all the absorbances is showing here. Okay. So that is how we can easily sum up our values. We don't sum up observances like I said earlier, but for the sake of demonstration, that is how we can sum up stuff using the editing, the AutoSum command. Okay, so that is it for today's overview of the Excel Home tab. Whether you are formatting chemical data, organizing experiment results, or calculating trends, the Home tab has all the tools you need to streamline your work. By mastering these features, you can spend less time formatting, more time analyzing your data. So, thank you for watching. If you found this tutorial helpful, don't forget to like and subscribe. And let me know in the comments what scientific Excel topics you would like to see next. See you in the next video.